Okay, welcome to video part 3 for this chapter. We'll start with blood. Basically how blood is composed, like what blood's made out of, is it can be separated into two types, the formed elements and the non-formed elements. Formed elements are cells. Non-formed elements is things that aren't cells. So you have red blood cells, white blood cells, and then platelets are kind of like cells. They're denucleated cell fragments of megakaryocytes, which, yeah, they have a mitochondria and things, but they just don't have a nucleus. Now, plasma is the other component of blood, and that's mostly made out of water, and there's also some proteins and antibodies as well. If you look at this graph right here, you'll see kind of the layout. You have alpha, beta, and gamma globulins and particles that you have in the plasma. The biggest negatively charged protein here is albumin. This is because it's most attracted to the positive charge, right? So it's over this way more. That's albumin, a really big component of it. And then alpha and beta proteins or globulins are just the complement system and the coagulation cascade and things like that, clotting factors. This gamma globulins are mostly antibodies, and so you see that they're closer to the negative end, which means they're positively charged, and that can affect ESR, right? And so if there's more gamma globulins, which can be caused by sickness or inflammation, the ESR will be higher. Now, in red blood cells, the most important substance really is hemoglobin, right? Red blood cells don't have mitochondria. They don't really produce energy. Their sole function basically is to carry oxygen. And so that's what hemoglobin does. Hemoglobin is made out of 4 globulin groups. And then within each of those globulin groups, there is also a heme group. And the heme group can attach to iron, and that's what allows it to carry oxygen. Now, the way that red blood cells can get broken down in our body, because they'll eventually get old. When they get old, the red blood cells, usually they're biconcave, right? And so that allows them to be more flexible and have more surface area for oxygen exchange. But as they get older, they become a little bit less flexible, and so they won't be able to fit in between these little gaps in the spleen. When that happens, the spleen, these little strands will kind of catch them. And then a bunch of why am I spacing the word? A bunch of white blood cells. They start with an M. They can phagocytose those red blood cells. So they'll kind of just like crawl along these and then start to eat and break down the red blood cell. And when it breaks down the heme, it'll break down the globins into amino acids, and then the heme will get broken down separately into the heme group and the iron. And what it'll do is the iron will then get picked up by a protein called transferrin, and that will be sent back to the bone marrow, so it can be incorporated into more heme groups and more red blood cells again. Now, the heme has a pretty special degradation pathway. What happens is the oh my goodness, what are they called? They are called macrophages. Okay. The macrophages will then break down the heme further. And first what happens is they turn the heme into biliverdin. And what biliverdin, there's an enzyme that does that's called heme oxygenase. And biliverdin's actually blue. And bilirubin, I believe it is soluble. It's kind of cool, so it'll kind of just keep flowing around. And then there's going to be another enzyme called biliverdin reductase around there that then turns it into bilirubin. Now what happens is bilirubin can twist around because of the pi bond that's broken, and so that will allow it to make hydrogen bonds with itself, and so it's not water soluble anymore. So what happens is it needs albumin to be able to carry it in the bloodstream. So it combines the albumin in the bloodstream, and then the albumin will carry it to the liver. Then inside of the liver, the bilirubin will be turned into conjugated bilirubin, which means there's going to be glucuronic acids which are added, and that's then soluble, and then that will be excreted as bile, with the bile, into the intestines. Now, in the intestines, there's some cool bacteria that will kind of eat away at the sugars, right, the glucuronic acids, which will cause the bilirubin to turn into urobilinogen. And urobilinogen is lipid soluble, and so there's a couple things that it can do. It can either return to the blood, and if it returns to the blood, it can have two pathways. It can be returned to the liver, which will then turn it back into conjugated bilirubin, and it'll kind of go through the cycle again, or it can be turned into urobilinogen in the kidney, or urobilinogen will turn to urobilin in the kidney, and that's what will be excreted as pee. It could also be turned into stercobilin. It'll be continued to break down by the bacteria in the intestine, and that is a brown color substance, and so that's what is excreted as feces. Okay, great. Now on to clotting. Now, a little bit of anatomy that we kind of need to know is there are some. You have the endothelial walls of the blood vessel, right? And then underneath it, there's collagen. And this collagen will be exposed if there's a break in the blood vessels, right? If you get a cut or something, blood starts to rush out, then there will be a break in that arterial wall. And these platelets that are floating around in the blood will then be attracted to the collagen. And these endothelial cells actually excrete what's called VWF. And VWF, von Willebrand factor, will bind to platelets. And then it'll also bind to collagen. And what happens is if it's bound to collagen and then a platelet comes along that then binds to it as well, if that platelet gets pulled, because the blood's moving this way, right, and so the platelet will get pulled, that will cause a conformational change and kind of activation of the platelet. And then the platelet will kind of turn into this crazy little guy that gets a lot bigger and kind of nastier. And what happens is an activated platelet, which gets activated because of VWF, which binds to collagen, right? So because of exposure to collagen, the activated platelet will first express fibrinogen receptors, which will be important. And then it will also start to release some substances inside of the cell. Now you have dense granules and alpha granules that will excrete substances. Dense granules will primarily excrete ADP and calcium. Alpha granules will excrete more VWF, which will help activate more platelets. And it'll also excrete PDGF, platelet growth factor, and that will allow for long-term healing and kind of allow the wound to be healed. It also does a lot of other things, including some factors in the coagulation cascade, but we won't talk about that right now. ADP, an important function of ADP is that it'll actually go and bind to receptors that are on platelets as well, which will then cause them to be activated. And then activated platelets will also start to produce this sort of this kind of phospholipid structure called thromboxane. Now, thromboxane will also serve to go and activate more platelets. Okay. Now, the receptors that we have on these cells include GP1B, which is the receptor for the Van Willebrand factor. There's the ADP receptors, and then there's also TXA2 receptors, which is for thromboxane. And that's kind of how they get activated. Great. Now what happens is this little platelet has expressed fibrinogen receptors, right? And fibrinogen is kind of just floating all around here in the blood. And when it will attach here, and then as another platelet gets activated by the ADP thromboxane or VWF, then it will also express fibrinogen receptors, and it will bind to the platelet. And so you can imagine there's just going to be a bunch of platelets bound to each other. And that's what we call platelet aggregation. And so a bunch of platelets will start to bind to each other, and that will kind of help start to form the clot and prevent blood flow out, kind of block that area. Now, on top of all of this that is happening, there's also what we call the clotting cascade. And there's two ways that we can kind of get into the clotting cascade. There's an intrinsic pathway and an extrinsic pathway. The intrinsic pathway, when there is activated by collagen. So when there's exposed collagen, that will activate factor 12. So factor 12 will get activated. And then factor 12 will activate factor 11, which will then activate factor 9. And factor 9 will kind of form a complex with a bunch of other things like factor 8 and calcium and phospholipids. And all this together will then form an activation complex for 10. And 10 is where the common pathway starts. So that's kind of where intrinsic and extrinsic combine. So this is the intrinsic pathway, right, activated by collagen, goes to 12. The extrinsic pathway, if there's damage underneath the endothelial cells, there's also some other cells, connective tissue and things that have TF, which is tissue factor, or also known as thromboplastin. And that's also known as factor 3. And what happens is factor 3 will cause activation of factor 7. And then factor 7 will form a complex with some other things like calcium and phospholipids, and I believe factor 5. And that will then go and activate 10. Great. So that's the intrinsic and extrinsic pathway. Once we reach the 10, then we call it the common pathway because both pathways converge here and they do the same thing now. 10 will activate 2. And 2 is actually called prothrombin, and then when 2 is activated, it becomes thrombin. And what thrombin does is it cleaves fibrinogen, or factor, also known as factor 1, and it turns it into fibrin. Now, what fibrin does is it will kind of it'll kind of form a weaving structure that will go around the clot and cause it to be held in place, and also to just connect everything all together. Now, thrombin will also activate factor 13, which will also help to stabilize the clot. Okay. And then what will happen is the clot will be all together, right? And there's going to be a bunch of fibrin as well going around here and kind of holding everything together. And then what will happen is the platelets will be connected to both ends of the break, and they have actin and myosin in them, and they'll contract, which will force the clot to pull together, and then that will hopefully start to heal this area. Now, I told you that fibrin kind of helps form that clot, right? Well, the thing that destroys fibrin is called plasmin. and that will get rid of fibrin. And so over time, you do want that clot to break down, and so your body will start to produce more plasmin. And the way that it does, one way that it does that is it produces TPA, which is responsible for cleaving plasminogen, which will then turn into plasmin. And that will cause the blood clot to slowly go away. Yeah. Now there is one other thing that contributes to hemostasis, also known as the cessation of bleeding, right? And one way that we can stop bleeding originally at like the very beginning is by constricting the blood vessels initially. And that will reduce blood flow, which will prevent more blood from leaving. And so that is a very temporary response and kind of primary response that happens right away. Just causes activation of vasoconstriction, which will cut off blood circulation for a little bit, but it is not permanent, and you need these platelets and this clotting cascade to make it all work out. Yeah, our body's pretty incredible.